So in order to understand how skeletal muscles contract, we need to understand the protein machinery that's behind that contraction or that shortening. And the topic of this segment is going to be a structure in the skeletal muscle cell that houses that protein machinery. That structure is called the sarcomere, and so the topic of this segment will be the sarcomere. Okay. So in order to understand where the sarcomere is and what it does, we have to go back for a second to the myofibrils. So this is where the muscle contraction is occurring. That is, the myofibrils are the organelles that are actually responsible for generating the pulling or the contractile forces in skeletal muscles. Okay, so we've got here our skeletal muscle cell or our skeletal muscle fiber, whichever term you prefer there. And recall that very densely packed within the skeletal muscle cell are these long rod-like organelles. We've got one of them pulled out here so you can see the detail of it here. We've got these long rod-like organelles that are very densely packed within the skeletal muscle cell. And these organelles are the myofibrils. Okay, so the myofibril is the contractile organelle for the cell. That is, this is the structure within the skeletal muscle cell that's generating those pulling forces and generating movement. Okay. The myofibril is actually made of thousands of small repeating units and that are essentially laid end to end, so that these are essentially strung end to end on the myofibril. And these repeating units are the sarcomeres. So this is where the sarcomeres are located. Okay. The sarcomeres actually contain two proteins that overlap, and these proteins interact with one another during shortening to actually generate the pulling force that we see in skeletal muscle. Okay, so that's where we're going to look. We're going to look at the sarcomeres, and we're going to look at the two proteins in these sarcomeres that interact to generate the contraction or the pulling force that we see in skeletal muscle. So let's take a closer look at the myofibril and at the sarcomere itself. Okay, so we've got our single myofibril here, and we're taking a look at a closer view, a closer examination of the myofibril itself. So the sarcomere is actually located between two structures or two borders that are called Z-discs. And so we have one Z-disc that's located here. We have the other Z-disc located here. And so everything between these two Z-discs is the sarcomere. So essentially these two Z-discs define the sarcomere itself. Okay, so this is one sarcomere. We've got another sarcomere here because this Z-disc also forms the border for this adjacent sarcomere to the left. This Z-disc forms the border for the sarcomere to the right here. And so everything between these Z-discs represents one sarcomere. The abbreviation Z here is actually a German word that I don't know how to pronounce because I don't speak German. So otherwise I would give you the full name of the Z-disc, but we'll just leave it at that. So we'll just leave it at Z-discs. So as I mentioned, it's within this sarcomere here that contains the two proteins that overlap and interact in order to shorten the muscle during muscle contraction. Okay, so that's what we're going to take a look at now. So when a muscle actually contracts, what is happening at the level of the sarcomere is that the two Z-discs are essentially being pulled closer together. That is, they're being pulled closer to one another. So here we've got a relaxed sarcomere here. This is the sarcomere of a muscle, a skeletal muscle, that has not been stimulated to contract. And here we've got the sarcomere after that skeletal muscle has contracted. Okay, that same sarcomere here. So essentially what happens is these two proteins, these two overlapping proteins that I just mentioned here, this one in red and this one in blue, they interact in a way that's going to pull these two Z-discs closer to one another. That is, they're going to pull them closer to the center of the sarcomere. And what that's going to do is it's going to cause the sarcomere itself to shorten, and because there are thousands and hundreds of thousands of sarcomeres end to end in the myofibrils of skeletal muscle, when you shorten all these sarcomeres or a lot of these sarcomeres simultaneously, what you're going to get is overall shortening of the muscle itself, and that's what's going to generate those pulling forces that we see. Okay. So not only does shortening require, or muscle contraction require, these two proteins overlapping and interacting with one another, it also requires these two substances here. It requires ATP. So these, particularly this substance here in blue, requires ATP in order to actually generate the pulling forces that are moving this red one here and moving the Z-discs. And also, calcium is a required substance that allows these two proteins to even interact in the first place. So we'll see the roles of ATP and calcium later on in muscle physiology. But just know for now that these are two substances that are critical in muscle contraction. Okay, without either of those two substances, the muscle would not contract. Okay, so these two overlapping proteins that I mentioned, we collectively call both of them myofibrils. Okay, myofilaments, not myofibrils, myofilaments. So myofibril is the contractile organelle. So again, there's that prefix myo that designates a muscular structure here. So these two structures are collectively known as the myofilaments. Okay, and so these are proteins that, as I mentioned, they bind together. They bind to one another, and they will pull the two Z discs in this diagram here closer to the center of the sarcomere. And that's that pulling and then moving of the two Z discs closer together is what causes the sarcomere and the muscle to shorten. Okay, so we're going to take a look at these two proteins now and what their structures are. This protein here that's anchored in the Z disc and extends to the center of the sarcomere or towards the center of the sarcomere is a protein called actin, or at least it's a structure called the thin myofilament that contains a number of proteins that are called actin. So basically this thin myofilament is made up of many of these little actin proteins. Okay, so collectively we call those actin proteins the thin myofilament. In the center of the sarcomere is a myofilament that is extending from the center of the sarcomere and towards each Z disc called the thick myofilament, and the protein that composes the thick myofilament is a protein called myosin. So there's lots of these myosin proteins within this thick myofilament. Okay, so we'll take a look first at actin, the thin myofilament. Okay, the proteins that make up the thin myofilament I mentioned is a protein that's called actin, and actin is a small bead-shaped protein. So the actin, when we refer to the thin myofilament or the actin filament, what we're referring to is a number of these actin proteins essentially strung together in a strand. Okay, so essentially the actin or the thin myofilament is essentially two strands of actin proteins that have been essentially kind of twisted together or twisted around one another. So they take on this kind of helix shaped pattern here. And the actin filament is firmly anchored at one end in the Z disc. So essentially one end of this thin myofilament is anchored into the Z disc, and it's associated with the Z disc. Okay. And actin, that is the protein actin itself, all these little individual actin proteins that make up this actin or thin myofilament, have a little place on it for the other protein, the myosin protein, which is on the thick myofilament, to bind to it. So it's critical that these two proteins, actin and myosin, are able to bind and interact with one another to cause muscle shortening. And so it's not surprising that actin has a myosin binding site on it. Okay. So another structure that's associated with the thin myofilament, or another set of proteins that's associated with the thin myofilament, are these two. These are proteins that are called troponin and tropomyosin here. And what you're seeing is that troponin is essentially kind of a pillar. It's kind of holding up the tropomyosin filament. And the tropomyosin, it kind of winds its way around, kind of following the helix or the twisting pattern of the thin myofilament, and what it does, the tropomyosin actually blocks, so it's physically blocking the myosin binding site. And so when the muscle is relaxed, that is, when the muscle is not being stimulated to contract, the myosin binding sites on actin are blocked by this protein called tropomyosin here. And we call troponin and tropomyosin, we refer to them as regulatory proteins, because these two proteins are essentially determining when the myosin and actin proteins are allowed to bind to one another. Okay, so we've got troponin and tropomyosin. So hopefully that's clear. Okay. Another protein that's associated with the thin myofilament is a protein called dystrophin. And what dystrophin does, and it's not actually shown in this diagram here, but what dystrophin does is that the dystrophin protein actually physically binds and links the thin myofilament and the actin proteins to the sarcolemma. So it essentially provides stabilization and structure and organization to the sarcomere itself. Okay, so disease or any abnormality in the dystrophin protein causes improper arrangement of the sarcomere. And this particular disease is a disease called muscular dystrophy. So muscular dystrophy basically is a disease that results from the fact that the sarcomere is not properly arranged, and so muscles can't contract properly in muscular dystrophy. And the reason for that is because this dystrophin protein is not correctly formed or correctly shaped, and so it doesn't provide the proper structure and the proper organization to the sarcomere itself. Okay, so we finished with the thin myofilament. We'll now take a brief look at the thick myofilament. And the thick myofilament is made of a number of proteins that are essentially bundled together. And these proteins that are bundled together to make the thick myofilament are called myosin. Okay, and myosin, really kind of the best way to describe myosin is it's kind of a golf club shaped protein. So it has these two kind of spherical head regions. It's got kind of a little skinny neck region that actually bends like a hinge. And then it has these two tail regions that are kind of twisted around one another. So one myosin protein is essentially, or one myosin filament, I guess I should say, is two of these myosin proteins that are kind of bundled together. So you can kind of think of them like the myosin filament as kind of two myosin proteins, kind of two golf clubs that have been twisted together, twisted around one another. Okay. So the myosin filament is made of many of these myosin proteins, and typically what you get in the myosin filament or the thick myofilament is that the myosin heads essentially extend away from the thick myofilament in sort of a circular pattern or circular arrangement. So they essentially extend from the thick myofilament in all directions. Okay, and the myosin heads have essentially two little binding sites on them. So these are the structures that are actually going to bind to the actin that we saw in the previous slide there. So the myosin heads have a couple of binding sites on them. They have an actin binding site. So this is where they will physically interact and connect with the actin proteins that we saw in the thin myofilament. And they also have a small binding site at the base of the head region, kind of near this flexible neck or hinge region that I talked about. And that is an ATP binding site. So myosin is actually the protein that moves and generates essentially the pulling force that causes muscle contraction. And in order to move and do that, it requires ATP. And so there's a little spot on this in order for ATP to bind and be hydrolyzed, that is to be broken down, so that ATP's energy can be released to generate movement to the myosin heads. Okay. All right. And as I mentioned, the myosin, essentially where the myosin head meets the tail region here, there is essentially a flexible hinge region. And so it's in this region that the myosin will bend and cause movement of the muscle there. Okay. There's one other protein that I want to discuss in the sarcomere, and that's a protein that essentially it anchors itself in one end of the Z disc and extends toward the center of the sarcomere, and it actually goes toward this. It actually meets the center of the sarcomere here, and this is a protein called titin. And as I mentioned, this is a protein that extends from a single Z disc, extends essentially through the center of the thick myofilament and anchors in the middle of the sarcomere here. So one titin protein extends from one Z disc to the center of the sarcomere. So it's a very big protein. And the titin proteins or the titin molecules actually make up what's called the elastic filaments in muscle. So if you see a structure that's being pointed out in any of the diagrams in your textbook or your lab manual, that's called the elastic myofilament or the elastic filament. That's this protein here, titin. Okay. So essentially, one of titin's functions is to essentially help suspend or support the thick filament in the center of the sarcomere. So as I mentioned, each titin molecule or each titin protein extends essentially through the center of the thick myofilament here. So it's helping to suspend and hold the thick myofilaments in the center of the sarcomere here. Okay. So essentially one titin molecule spans half of the sarcomere width. And so because of this, titin is a very large protein. And that's actually why it's named titin, because titin implies that this is a huge protein. And actually, in fact, titin is one of the largest proteins that has been described in biology to date. So this is a very large protein. And you'll also notice that titin has this kind of essentially this kind of coiled, almost spring-like region here. And it turns out that one of titin's other functions is to actually give the muscle the property of elasticity. So remember I mentioned that you can stretch a muscle out and it will return back to its original length. And essentially just like a little rubber band. And this is the molecule, the titin molecule, imparts this elastic property to muscles, because essentially this little region here of titin essentially kind of behaves like a little spring. And so this is what allows titin to be, or this is what allows the muscle to behave like a rubber band. Okay. So let's take a look at the overlap of the thick and thin filaments within the sarcomere. So if you were to look at a light microscope image of a sarcomere, a very detailed light microscope image of a sarcomere, what you would see is that you would see these very characteristic alternating light and dark banding patterns. So what we got here, we have our diagram of our sarcomere here. So we've got our Z discs here. We've got our thin myofilaments in blue here, our thick myofilaments in red here. And you're seeing essentially an exact, that's exactly kind of overlaid on this, the organization of the sarcomere here. So what we have here are the two Z discs, and then everything within this is the center of the sarcomere here. And so the overlap of these thick and thin filaments creates this light and dark banding pattern within the sarcomere. So towards the center of the sarcomere, we have this very dark region, and this very dark region is referred to as the A band. And this is the region where the thin myofilaments, that is the actin filaments, overlap with the thick myofilament, right? So because this region is sort of very densely packed with proteins due to this overlap, this region in the sarcomere appears very dark under the microscope, and so we call that the A band. Okay, so it appears dark, so think the A is in dark, A is in A band, right? And then towards the end of each sarcomere, so essentially flanking each Z disc, spanning each side of the Z disc, is what we call the I band. And so this kind of lighter region of the sarcomere, this region here and this region here, which is kind of, these two light regions here are part of this focal sarcomere here, but these two light regions here are part of other sarcomeres that we can't see here. Essentially, these regions here are what are called the I bands, and they span each side of the Z disc. That is, they span the left and the right side of each Z disc. And the reason that they appear light in color is because there isn't the overlap of protein here. So that is, the proteins aren't as densely packed here, and so this causes these regions of the sarcomere to appear a lot lighter, because only thin filaments and only titin are seen here. Okay, and as I mentioned, it appears light under the microscope image. So think the I in light as in the I band. So how do these two proteins interact to cause the muscle to contract and shorten, right? So basically what happens is that, so recall that myosin, the myosin proteins have binding sites on them for actin, and the actin proteins have binding sites on them for myosin. Basically what happens is that myosin physically attaches itself to actin and it pulls. And when it pulls, because actin is anchored to the two Z discs, when myosin pulls on these actin filaments, it's also going to pull on the Z discs. And when these Z discs are pulled, they're going to move closer to the center of the sarcomere. And so essentially what this is going to do is it's going to shrink or decrease the distance between the two Z discs, and it's going to decrease the length of the sarcomere temporarily. And so, as I mentioned, you do this many hundreds of thousands of times with lots of sarcomeres chained end to end, and you're going to get an overall shortening of our entire muscle. So our Z discs are going to be pulled closer together, and our sarcomere is going to shorten. Okay. And this model, or the way that we describe the myosin pulling the actin filaments, is described as what's called the sliding filament model. And basically what happens is myosin attaches to the actin and pulls on it, and so actin slides over or glides over the myosin and moves the Z discs closer to the center of the sarcomere. So we call this model of muscle movement, muscle contraction, we call it the sliding filament model.